Hey guys, this is going to be the first video clip I'm sending you. I would suggest you draw this out with me. This goes with section 6.2, and this is about the replication of your DNA. When our cells divide, we have to replicate the DNA so we have twice as much of it. So when the cell divides, one of them will have half of that and the other one will have the other half of that DNA. Okay. So let's get started. We know DNA, we learned that is our genetic material that codes for all of our traits. It is a type of nucleic acid. And nucleic acids are made up of the building blocks called nucleotides. Okay? So we know that a nucleotide is a sugar. In the case of DNA, this would be ribose sugar, and a phosphate, and a nitrogenous, or nitrogen, let's say, base. And that nitrogen base could be adenine, that will just illustrate with an A. It could be guanine, that will illustrate with a G. Could be cytosine, we'll say with a C. Or it could be thymine. Let's put the T over here, because the T always pairs with the A and the G always pairs with C. We'll find that out later when we get a little bit more into DNA structure. So you've got all these building blocks that go together, and you know DNA is a double helix. It's kind of like that double ladder deal, and when these nitrogenous bases pair together, they're held with hydrogen bonds. Okay, so to simplify it, instead of drawing out the whole nucleotide, we'll make a strand of DNA with just the letters. And anytime you've got A, it pairs with T. If you got a C, it's going to pair with a G. Say another T, it'll pair with an A. Say another G, it'll pair with a C, and so forth. Well, there is a enzyme called DNA helicase. We see it ends in ASE, so it's got to be an enzyme. And that comes in like a pair of scissors right here and starts snipping those hydrogen bonds. And as it cuts them, it's making one strand of the DNA on this side, the other strand of the DNA on that side. So we'll just keep putting that out here. A, C, T, G, and on this other side we've got T, G, A, C. Now there's another enzyme called DNA polymerase. And these polymerase enzymes come in and start building the nucleotides that match up with these old strand nucleotides. So everywhere there would be an A, it would put a nucleotide with a T. Everywhere there's a C, it would put a nucleotide with a G. With a T, it would be an A. Where there's a G, it would put a C. And on the other side, another polymerase would be reading this side of DNA. Wherever there's a T, it would put an A, and a G, it would put a C, and an A, it would put a T, and wherever there's a C, it would put a G. And this is how our DNA is replicated. When you have a replication with an old side and a new side, this strand of DNA is going to be exactly like this strand of DNA. We would call that semi-conservative replication. Okay, so that is how your DNA replicates when the cell is getting ready to divide, by semi-conservative replication. The DNA helicase splitting the strands of DNA, and the DNA polymerase coming in and reforming new strands. So in the end of it, we have one old strand with a new strand, and on the other side, an old strand with a new strand that are exactly the same, and therefore your DNA has been replicated.